In this video, we will be demonstrating the process of pronuclear injection, including embryo collection, injection, and embryo transfer with mice. Pronuclear injection will be demonstrated using mouse, rat, and rabbit embryos. Step 1. Embryo collection. Begin by removing the oviduct and placing it in a small dish of media under the dissection scope. Tear the ampulla with a small syringe needle and pull out the embryos into the surrounding appropriate media. We recommend M2 medium for mice. Carefully add hyaluronidase to remove cumulus cells surrounding the embryos. After 5 minutes of incubation, the cumulus cells should dissociate and the embryos can be collected in a glass capillary tube and moved around the media to further clean them of cumulus cells. We recommend a solution that is 1 mg per ml hyaluronidase with 10 mg per ml polyvinyl pyrrolidone in 0.9% saline. Gather the cleaned embryos and place them in a new dish containing media for transfer to the microinjection station. If needed, repeat this step until embryos are free of debris. Collect the mouse embryos for pronuclear injection. Step 2. Pronuclear injection. Before we begin the microinjection, let's quickly review the injection apparatus and highlight the relevant anatomy of the single-cell embryos that we will be working with. The holding pipette and injection needle are on opposite sides of the embryo. The holding pipette is used to ensure that the embryo remains stationary while the injection pipette is inserted into the embryo. Now let's look at embryo anatomy. The embryo is surrounded by 2 outer membranes, the zona pellucida and vitelline membrane. Between these 2 membranes lie the polar bodies, which are the byproduct of the uneven cell division that occurs during meiosis. Inside the embryo, we have the pronuclei surrounded by pronuclear membranes and containing nucleoli. We will be injecting our ZFN reagents directly into one of these pronuclei. Begin the injection procedure by using the microscope coarse focus knobs and pipettes to orient the embryos so the pronucleus is in an accessible position on the right side of the embryo. This is done by expelling pressure out of the holding pipette and applying suction, using the tip of the needle as an anchor. Once the pronucleus is in focus, move the injection needle either up or down to bring the tip of the needle into focus. Having both the pronucleus and the tip of the needle in focus should assure that both are in the same viewing plane, allowing the needle to directly penetrate the pronuclear membrane. Next, move the injection needle into the embryo, making sure to move in a planar fashion, piercing the zona, the vitelline membrane, and the pronuclear membrane. Once the pronuclear membrane is pierced, expel the injection solution into the pronucleus. A 10% swelling of the pronucleus should be observed, and the pronuclear membrane will become much more defined. Carefully remove the needle out of the embryo. Pronuclear injection is usually performed on the male pronucleus because of its larger size. However, if the male pronucleus is in a difficult spot, then the female pronucleus can be injected successfully. The mouse pronuclear membrane is quite rigid, making it fairly easy to penetrate. The rat pronuclear membrane is a little more pliable than the mouse pronuclear membrane, requiring the needle to travel farther past the pronuclear membrane or requiring application of a little popping motion by the injectionist to pierce the membrane. Once the pronuclear membrane is pierced, expel the injection solution into the pronucleus. A 10% swelling of the pronucleus should be observed and the pronuclear membrane will become much more defined. Carefully remove the needle out of the embryo. The rat embryo is similar in size to the mouse embryo, with a diameter of about 100 microns. However, the rat pronuclear membrane is more pliable and is more difficult to pierce. The rabbit embryo is much larger in size than either the mouse or rat, with a diameter of about 150 microns. Additionally, the rabbit zona and vitelline membranes are thicker and more difficult to enter than the mouse or rat membranes, requiring a sine or piezo motion by the injectionist to pierce. Begin the injection procedure by using the microscope coarse focus knobs and pipettes to orient the embryo so that the pronucleus is in an accessible position on the right side of the embryo. This is done by expelling pressure out of the holding pipette and applying suction, using the tip of the needle as an anchor. As the zona and vitelline membranes are thicker and more difficult to enter than the mouse or rat membranes, a sine or piezo motion is required by the injectionist to pierce them. Once the pronuclear membrane is pierced, expel the injection solution into the pronucleus. A 10% swelling of the pronucleus should be observed, and the pronuclear membrane will become much more defined. Carefully remove the needle out of the embryo. In this example, once the pronuclear membrane is pierced, the embryo shifts when the needle enters, causing the pronucleus and the needle to be in different planes. The microinjectionist removes the needle and refocuses on the pronucleus and then reinserts the needle. Care should be taken when working with embryos to ensure viability. In the next couple of examples, the injectionist will demonstrate situations that should be avoided. The microinjectionist purposely pierces the zona and vitelline membrane and moves the needle incorrectly, and also moves the injection needle in and out of the embryo multiple times. You can see the cytoplasm start to leak out into the zona space. This leakage will cause cell lysis and inevitably apoptosis to occur. For illustration purposes, the microinjectionist purposely pierces the zona and vitelline membrane and presses the purge function of the femtojet, which expels an enormous amount of injection solution out of the needle. DNA in the cytoplasm alone can cause apoptosis. This large amount of solution injected into the embryo causes the pronucleus to move out of the cytoplasm slightly, and cell death will occur. The embryo lysed by purging is compared to previously injected embryos that are healthy. This is to show a lysed, dying embryo compared to a non-lysed, living embryo. Embryos can differ greatly across species, and while mouse and rat embryos have similar compositions and sizes, the rat embryo has tougher membranes. Rabbit embryos have much tougher membranes and are roughly 2.5 times larger in size compared to rat embryos. Step 3. Embryo transfer. When pronuclear injection is complete, transfer the embryos back to the dissection scope for transfer into anesthetized pseudopregnant recipient animals. If multiple slides will be injected, the embryos may be pooled into pre-equilibrated media drops overlaid with oil and held in the incubator until all embryos are injected. Begin the embryo transfer process by collecting the embryos to be transferred in a glass capillary tube. Embryos should be transferred into the exposed oviduct. Begin by orienting the exposed organs to facilitate access to the infundibulum. Tear the bursal membrane and expose the infundibulum. Insert the closed end of a fine pair of forceps to confirm proper orientation and access. With the infundibulum exposed, insert the glass capillary containing the embryos to be transferred and eject them completely into the oviduct. The injection of air bubbles behind the embryos is used as confirmation of a successful embryo transfer. After successful transfer of the injected embryos into the oviducts of the pseudopregnant recipient, close the incisions using suture and/or wound clips. Place the animal in a solitary cage that has been warmed using a heat pad, and allow the animal to recover. Our mice are generally multiple housed up to 5 per cage until about 4 days before the due date. At this point, they are broken into cages containing 1 to 2 females. We target 4 recipients per session of injection. Please follow your institution's established guidelines regarding proper animal husbandry. Sigma-Aldrich, enabling science to improve the quality of life.